Welcome to EPG Pathshala. I am Dr. Janvi Manhas from the Department of Biochemistry, All India Institute of Medical Sciences. Today we are going to discuss about the module PCR, that is polymerase chain reaction and its applications. This module is placed under the paper 11 Cellular and Molecular Biophysics. Let us start the topic of polymerase chain reaction. Polymerase chain reaction is an enzymatic method of amplifying a particular sequence of DNA many folds in vitro. That is, this amplification takes place outside in a test tube. As we can generate millions of copies of DNA in this technique is very useful for molecular diagnostics, forensic science and in medical research such as cancer research and drug discovery. In this module, we will be understanding the basic concept of polymerase chain reaction and the 3 steps involved. We will also discuss the instrumentation and different components of polymerase chain reaction or PCR. In addition, we will discuss reverse transcriptase PCR and real-time PCR. Finally, we will enumerate the wide variety of applications of PCR in medical research and diagnosis. The basic idea of replicating a piece of DNA using 2 primers has already been described by Khurana in 1971. But the progress was limited at that time due to the inability to synthesize primers, and there was also purification issues related to the DNA polymerase. PCR was first developed in early 1980s by the American biochemist Kary Mullis. The Nobel Prize in Chemistry 1993 was awarded for contributions to the development of methods within DNA-based chemistry, jointly with one half to Kary B. Mullis for his invention of the polymerase chain reaction method and with the other half to Michael Smith. Michael Smith was awarded for his fundamental contributions to the establishment of nucleotide-based site-directed mutagenesis. PCR is now a widely used molecular technique used in medical and biological research labs for a variety of applications. Once amplified, PCR products, also known as amplicons, can simply be visualized by agarose gel electrophoresis. They can also be cloned, sequenced or be subsequently digested with restriction enzymes for DNA fingerprinting. In April 1990, Scientific American published an article written by Kary Mullis, which was titled The Unusual Origin of the Polymerase Chain Reaction. This article gave an account of the discovery of PCR. Highlighting the importance of PCR, Mullis wrote, beginning with a single molecule of a genetic material DNA, the PCR can generate 100 billion similar molecules in an afternoon. The reaction is easy to execute. It requires no more than a test tube, a few simple reagents and a source of heat. Now let us discuss the simple technique of PCR, which is very useful in molecular diagnostics. PCR is a quick and inexpensive way to create a number of copies of a single DNA segment. Now let us understand this PCR or polymerase chain reaction in detail. PCR is a quick and inexpensive way to make multiple copies of a targeted DNA. These multiple copies of a specific DNA can be formed from a very minute quantity of starting material. There are 2 basic reasons to amplify a DNA segment. The first is to create multiple copies of rare DNA. Now, what is rare DNA? Let's take an example of a forensic science scenario in which we have evidence as a single drop of blood or a single hair. In order to help in the criminal investigations, we need to amplify this small amount of DNA from a small hair or a drop of blood, and in this PCR is a useful technique. The second reason is to compare 2 samples for abundance of DNA. Comparing two samples for abundance of DNA can help us understand which sample has more expression of this DNA. Now, PCR is a technique very similar to DNA replication. In this technique, a double-stranded DNA is first unwound into single-stranded DNA. Then specific oligonucleotide sequences known as primers attach to this single-stranded DNA. And with the help of DNA polymerase, extension of these primers occurs, which is complementary to the single-stranded DNA. This leads to amplification of the DNA. Because PCR is so similar to DNA replication, let us have an overview of DNA replication. The replication of DNA is the process on which PCR amplification is based. During replication, the DNA molecule unwinds, with each single strand becoming a template for synthesis of a new complementary strand. Each daughter molecule consists of one old and one new DNA strand and is an exact copy of the parent molecule. DNA polymerase is a very essential and useful enzyme here. It can add nucleotide onto a pre-existing 3' OH group only. Therefore, it needs a short DNA sequence or primer to which it can add the first nucleotide. DNA polymerase also requires a supply of DNA building blocks, that is, the 4 bases: adenine, that is A, thymine, that is T, cytosine, that is C, and guanine, that is G. Just as the replication of DNA takes place, amplification takes place in a PCR reaction. In PCR, one DNA molecule is used to produce 2 copies, then 4, then 8, and so on and so forth. Because both strands are copied during PCR, this results in an exponential amplification of the target region of DNA. This continuous doubling and cycling is made possible by the use of Taq DNA polymerase, a versatile thermostable DNA polymerase isolated from the bacterium Thermus aquaticus. At the end of the PCR reaction, the specific target sequence is accumulated in billions of copies. In 30 rounds of replication, 2⁻30, that is, more than a billion DNA molecules of the target gene are produced for every starting double-stranded DNA molecule. This is akin to a molecular photocopy of a target DNA, specifically selected by designing primers against the flanking DNA sequence of the target DNA to be photocopied or amplified. Now, since we have been comparing the PCR, that is polymerase chain reaction, with DNA replication, let us see some of the differences between the two. First of all, the place where the DNA polymerase attaches to the DNA strand is an RNA-DNA hybrid in vivo replication. However, in PCR, it is a DNA-DNA hybrid. For separation of the DNA strands of the double-stranded DNA, helicase enzyme is used in vivo replication. However, with the application of heat, we are able to separate the double-stranded DNA in PCR. The enzyme that elongates new strand of DNA is a thermolabile DNA polymerase in vivo DNA replication. However, we use a thermostable Taq polymerase for PCR reaction. The primers in vivo replication are made of RNA, but we synthesize artificially oligonucleotide sequences of DNA in PCR. Now let us learn the essential steps of PCR. There are 3 essential steps of PCR: denaturation, annealing, and extension. We will discuss all of these 3 in detail. The purpose of PCR, that is polymerase chain reaction, is to make a huge number of copies of a desired DNA. This is done in an automated cycler which can heat and cool the tubes with the reaction mixture in a very short time. The first step, that is the denaturation, takes place at about 94 degrees centigrade, and in this step the double-stranded DNA melts and opens into single-stranded DNA. The second step, that is the annealing, takes place around 54 degrees centigrade. The primers that are moving around in the reaction mixture form ionic bonds with the separated single-stranded DNA. This process is known as annealing. Once a few bases are built in, the ionic bond is so strong between the template and the primer that it does not break anymore. This leads us to the third important step, that is extension. Extension takes place at 72 degrees centigrade. The primers which are attached to the single-stranded DNA are acted upon by DNA polymerase, which is a thermostable DNA polymerase. The DNA polymerase adds DNTPs or nucleotides from 5' to 3' direction and extends the fragment, thereby leading to amplification of the DNA fragment. After the PCR amplification has taken place, we have to check whether the desired PCR product has been formed or not. There are 3 steps that have to be taken care of. The first is to check whether the product is formed or not. The second step is to check whether the product is of the right size or not. The third important step is to see if there is specific amplification. All these things can be achieved by running the PCR product on gel electrophoresis. In order to make sure that our PCR is successful, we should do a gel electrophoresis of the PCR product. In the next figure, you can see gel electrophoresis of different PCR products, and we are going to understand how to interpret the gel electrophoresis of PCR products. In this picture, you can see that there is a ladder. The ladder is a mixture of fragments with known size, and it helps us to compare and identify the PCR products. Lane 1 has PCR fragment of approximately 1850 bases long. Lane 2 and 4, the fragments are approximately 800 bases long. Lane 3, you cannot appreciate a band, that is, there is no product formed and the PCR reaction has failed. In lane 5, you can see multiple bands are formed. This can be due to non-specific primers which are amplifying different regions of the template DNA. Now let us understand and study the different components of a PCR reaction. We have already studied the 3 steps, that is denaturation, in which the double-stranded DNA separates into 2 single-stranded DNAs. The next step, annealing, in which the primers attach to the single-stranded DNA because primers have complementary bases to flanking regions of your desired DNA. And the third step, in which DNA polymerase acts and cause extension of these primers to cause amplification of the desired DNA. Let us understand the different components that are required for this PCR reaction. The first component is the DNA sample. The DNA sample has the template DNA. The second component are the primers. These primers are specifically designed towards the flanking region of your template DNA. The third component are the nucleotides. These are the third component are the nucleotides. These are the building blocks for DNA amplification. The fourth and very important component is Taq DNA polymerase. The fifth component is the mix of the buffer and divalent cations useful for the action of Taq polymerase. All these 5 components go into a reaction mixture in a PCR tube. This PCR tube is placed in a thermal cycler. Now let us see what is this thermal cycler. Now let us see what is this thermal cycler. A thermal cycler is the basic instrumentation required for PCR reactions. The thermal cycler is also known as PCR machine. This thermal cycler raises and lowers the temperature of the samples in the holding block in discrete pre-programmed steps, allowing for denaturation and re-annealing of samples with various reagents. In this picture, you can see an open thermal cycler or PCR machine, and you can appreciate the block in which the rise and fall of temperature takes place. PCR tubes are thin-walled plastic tubes which can be used for holding the reaction mixture in this block. Now let us discuss each component one by one and in detail. Starting with the first component, that is the starting material, DNA template. In this figure you can see DNA which is marked in red, and then there is a target DNA which is the DNA to be amplified. To target this DNA and amplify this DNA, primers are designed to the flanking region of this DNA. So we need a good quality starting material, that is DNA template for PCR reaction. On the left hand side, you can see a spectrophotometric analysis of the DNA sample for PCR reaction. This analysis is based on the principle that nucleic acid absorbs ultraviolet light in a specific pattern. In the case of DNA and also RNA, a sample exposed to ultraviolet light at a wavelength of 260 nanometers and a photodetector measures the light that passes through the sample. Using this method, we can measure both purity and concentration of template DNA, which is to be used for amplification in PCR. So this is the double-stranded template DNA, and during the first step of the PCR reaction, when the temperature of the thermal cycler is 92 to 94 degrees centigrade, the double-stranded DNA, as you can see, melts, that is, separates into single-stranded DNA. Now let us discuss about the second important component, that is the primer. A primer is a short synthetic oligonucleotide designed to have a sequence which is the reverse complement of a region of the template DNA to which we wish the primer to anneal during the PCR reaction. Primer designing is very important for a PCR reaction, and there are certain criteria to be followed. Generally, the primers used are 18 to 25 nucleotides in length that allows the use of a reasonably high annealing temperature. Primers should avoid stretches of polybase sequences or repeating motifs. These can hybridize inappropriately onto the template. Also, we should avoid inverted repeat sequences so as to prevent formation of secondary structure in the primer. Sequences complementary to other primers used in the PCR should also be avoided. If possible, the 3' end of the primer should be rich in G and C bases to enhance annealing of the end that will be extended. Why the primer length of at least 16 nucleotides is important? We know that there are 4 bases in the DNA molecule: adenine, cytosine, guanine and thymine. The probability of encountering any of these bases in the code is 1/4, that is 0.25. So let us look at the possibility of encountering a particular sequence of bases. When only one base is used, the probability of encountering it is 0.25. As the number of bases increases, the probability becomes 0.25 raised to power the number of bases. So when we use 16 nucleotide primer length, it becomes increasingly unlikely that one will get 16 bases in this particular sequence in the DNA template. In the same way, one can see that as the primer increases in size, the chances of a match other than the targeted one is highly unlikely. By this explanation, longer the primer, more chance of being unique and highly specific. However, we still have a cap. We cannot go beyond 30 bases. Let us see why. So what would happen if the primer length is too long? If the primer is too long, the annealing temperature will be too high. The chances of self-complementarity will be higher and secondary structure formation will be higher. All these conditions will lead to failure of PCR. Hence, the primer length is usually recommended to be 18 to 24 nucleotide long for human DNA sequences. This brings us to the second step, which is annealing. As we have seen the different requirements for primer designing, after designing a specific primer at 55 to 65 degrees centigrade, the primers bind to the complementary sequences of denatured or melted DNA, and this process or step is known as annealing. Coming to the third component of PCR, that is the Taq polymerase. Now, Taq polymerase was a big leap forward in PCR. Before Taq polymerase was discovered, the DNA polymerase used by Kary Mullis for PCR reactions was thermolabile. So the PCR reaction or PCR technique was not that simple. It was extensively elaborate and required a long time. As you can see in this picture, there are 3 water baths maintaining different temperatures because at that time the thermal cycler was also not available. Also, the DNA polymerase was thermolabile, that is, once when the 95 degrees centigrade denaturation step was achieved, the DNA polymerase would degrade. Therefore, after denaturation in each cycle, DNA polymerase had to be supplemented afresh. To tide over this problem of labile DNA polymerase, thermostable DNA polymerases were discovered. These thermostable DNA polymerases were derived from Thermus aquatis, which is an organism of the hot springs of Yellowstone National Park in USA. Besides Taq polymerases, there are other DNA polymerases which are thermostable. Now let us see what is the difference between Taq polymerase and PFU DNA polymerase, which is another thermostable DNA polymerase. The organisms from which these two polymerases are derived are different. That is, the Taq polymerase is derived from Thermus aquaticus, whereas the PFU is derived from Pyrococcus furiosus. The extension rates are also different. Taq polymerase extends the primer at a rate of 2 kilobases to 4 kilobases per minute, whereas the PFU DNA polymerase extends 1 kilobase to 2 kilobase per minute. The half-life at 95 degrees centigrade, that is the denaturation step, is very high for PFU DNA polymerase, that is 4 hours as compared to Taq DNA polymerase. The processivity is high for Taq DNA polymerase, that is 50 to 60 bases. The 5' to 3' exonuclease activity is present in Taq DNA polymerase, whereas in PFU DNA polymerase 3' to 5' exonuclease activity is present. Let us discuss about Taq DNA polymerase, which is a thermostable DNA polymerase and very frequently used for PCR reactions. The Taq DNA polymerase has a half-life of 45 minutes at 95 degrees centigrade. The extension rate is 2 to 4 kilobases per minute. Processivity is 50 to 60 bases per molecule, and the optimum temperature and pH is 72 degrees centigrade and 7.2. Now, what is the reason for the stability of this Taq polymerase? There are certain properties of this enzyme which make it thermostable. The 3 important properties are increased hydrophobicity of the core of this enzyme, increased disulfide linkages and more proline content. Owing to these 3 properties, the DNA Taq polymerase is stable at very high temperatures of 95. Let's discuss the next 2 components of PCR reaction, that is the nucleotides and the reaction mixture. So nucleotides are very important for a PCR reaction because they are the building blocks of DNA. We generally use equimolar concentration of nucleotides. The 4 different types of nucleotides, that is D-ATP, D-CTP, D-GTP and D-TTP. The buffer used is tris hydrochloride buffer, which maintains a pH between 8.3 and 8.8 at room temperature. However, during a PCR reaction, when the temperature is 72 degrees centigrade, it maintains the optimum pH of 7.2, which is required by the thermostable DNA polymerase. Also, this reaction mixture contains the very important divalent cation, that is magnesium. This divalent cation is very useful and essential because it is an important cofactor of the DNA polymerase. Also, this divalent cation, that is magnesium, helps in the incorporation of the nucleotides in the growing end of the DNA. It also increases the TM of the primer and template hybrid so that these do not separate during the extension step. The extension step, as we have discussed, occurs at a temperature of about 72 degrees centigrade. It is required for 30 to 60 seconds. During this step, you can see in this figure that the purple box marked as Taq polymerase binds to the annealed primers and extends the DNA at the 3' end of the segment. In this figure, you can see a typical PCR run as shown in a thermal cycler. You can see the initial denaturation step of DNA, that is 95 degrees centigrade for 3 minutes, followed by the 3 essential steps of PCR reaction: denaturation, annealing and extension. These 3 steps go on in a cyclical manner for 30 to 35 cycles, at the end of which there is a hold step to preserve the PCR products at 4 degrees centigrade. So, starting with a single copy of DNA and depending upon the number of cycles the PCR reaction occurs for, the copies of DNA at the end of the PCR reaction is equal to 2 raised to power n, where n is the number of cycles. Just to rewind and give you an overview of whatever we have learned till now, let us see a short movie of PCR reaction as it is happening in the thermal cycler. Inside the thermal cycler, which can heat and cool the tubes with the reaction mixture in a very short time, all 3 steps are happening. The first step is the denaturation at 94 degrees centigrade, where the double-stranded DNA melts into single-stranded DNA. The second step is annealing, as you can see in the movie, at 54 degrees centigrade, where the primers which are moving around in the reaction mixture anneal with the single-stranded DNA. The third step is the extension, which occurs at 72 degrees centigrade, wherein the primers are extended by DNA polymerase from 5' to 3' direction. We have already discussed that at the end of the PCR reaction, the number of DNA copies is equal to 2 raised to power n. That is, n is the number of cycles. Now, what does this mean and why should we restrict ourselves to 30 to 35 cycles of DNA only? Let us understand this by the next example. In this figure, you can see that as the cycle number increases, the DNA copy number increases exponentially. So, there is exponential expansion from cycle number 1 to cycle number 30. However, after cycle number 30, the ability of exponential expansion or exponential amplification is lost. Also, you can see the 2 graphs on the right where the DNA copy number is plotted against the PCR cycle number, and you can appreciate in both of these graphs that after cycle number 30, the exponential amplification does not happen. Let us understand why. To understand the limiting factors after 30 or 35 cycles of PCR, let us understand the phases of a PCR reaction. There are 3 phases of PCR amplification: exponential, linear and plateau. The exponential phase is the first phase of PCR amplification. The reaction components in this phase are in excess. There is an exact doubling of product in each cycle and the reaction is specific and precise. There is a special type of PCR known as real-time PCR, which we will discuss in a few slides. This real-time PCR measures the cycle number value at this phase of PCR. The second phase is the linear phase of PCR amplification. The reaction components in this phase are being consumed. Therefore, the amplification slows down and the reaction becomes highly variable. The final phase of PCR amplification is the plateau phase. The reaction is complete and no more products can be generated. Also, the limiting factors here are the DNTPs, the enzyme as well as the other factors. Traditional PCR takes measurements during this phase of the PCR reaction. This is one of the limitations of PCR. The other limitations of conventional PCR include size-based discrimination only, which can be seen by doing a gel electrophoresis after the PCR run is complete. Also, it is an end-point detection assay. We cannot know the amplification during each cycle of the PCR reaction. The results are not expressed as numbers and we have to do a gel run to find out if our PCR was successful or not. The double-stranded DNA dye, which is utilized for quantification of conventional PCR, is ethidium bromide, and ethidium bromide staining is semi-quantitative. An improvement over the conventional PCR is the real-time PCR. Conventional PCR is an end-point assay. However, real-time PCR measures the PCR product formed real-time. That is, measurement is done after each cycle of the PCR. In real-time PCR, the amount of DNA produced after each cycle can be quantitated using a double-stranded DNA dye which binds only to double-stranded DNA. When the double-stranded DNA dye binds to double-stranded DNA, it emits a fluorescence which can be captured by the real-time PCR machine. We can also use oligonucleotide probes which can hybridize to the target DNA and give a fluorescence signal which can be detected after each cycle of the PCR. The cleavage of these hydrolytic probes is done by Taq polymerase, which is already utilized in the PCR reaction. The PCR mixture is prepared along with the double-stranded DNA binding dye, that is cyber green, or the hydrolysis probes, that is Taqman probe. After each cycle, the level of fluorescence is measured. The machine has fluorescence detectors which captures the signals and converts them to graphical representation on the screen. CT values, that is cycle number at which threshold fluorescence is achieved, are calculated by the software. With reference to a standard dilution, the double-stranded DNA concentration in the PCR can be determined. There are a number of quantification probes which can be used in real-time PCR or qPCR. The first one is the Taqman probe. Taqman name comes from Taq, that is Taq polymerase, and man comes from a video game, that is Pac-Man. The Taqman probe assay is one of the earliest methods introduced for real-time PCR reaction. The 5' endonuclease activity of Taq DNA polymerase cleaves an oligonucleotide probe during PCR, thereby generating a detectable signal. The probes, as you can see in the green color, have a 5' label with reporter dye and a 3' label with a quencher dye. The sequence of the probes is such that it will hybridize to the complementary templates. Proximity of the two dyes allows the quencher dye, that is the Q as seen in the figure, to absorb the emission of the reporter dye. During polymerization, the Taq enzyme displaces and cleaves the hybridized probes, producing an exponential increase in cleaved reporter dye emission at each cycle. We can also use DNA binding dyes for quantification in real-time PCR. One of the most commonly used DNA binding dye is cybergreen. Let us see how it works. Cybergreen is a DNA binding dye which binds to double-stranded DNA in a PCR reaction. It fluoresces only when bound to double-stranded DNA. An increase in double-stranded DNA product leads to an increase in fluorescence intensity, which is measured at each cycle, thus allowing template DNA amplification to be quantified in real time. There are some important terms in real-time PCR. Knowing the definition and understanding the importance of these terms helps in quantification of real-time PCR and in its measurements. Now, real-time PCR uses an increase in the intensity of a fluorescence signal generated by an intercalating dye or from the breakdown of a dye labeled probe during amplification of a target sequence to detect nucleic acids, either for the presence or absence or for their amount. As you can see in this graph, there is a baseline. This baseline is defined as the number of PCR cycles in which the reporter fluorescence signal is beneath the limit of detection of the instrument. The next important term here is the threshold. Threshold is an arbitrary level of fluorescence which is chosen on the basis of the baseline variability. Any signal detected above the threshold level is quantified and used to define the threshold cycle, that is CT value, for a sample. The threshold should be adjusted for each PCR run so that it is placed at the region of exponential amplification across all plots. The third important term here is the CT, that is threshold cycle number. CT is defined as the fractional PCR cycle number at which the reporter fluorescence is greater than the threshold. The CT is a basic interpretation of real-time PCR and essential to produce accurate and reproducible data. You can also see on the y-axis delta Rn. Now, this is an increment of fluorescent signal at each time point. The delta Rn values are plotted versus the cycle number to get the quantification plot. In this figure, you can see real-time quantification of different samples. You can see that the sample with the highest number of template DNA has the lowest cycle number. That is, the curve rises earlier if the initial template number is high. You can see the purple plot which has only one DNA copy to start with. So, exponential amplification occurs at very high cycle number for very low template DNA. A very important aspect of real-time PCR quantification is melt curve analysis. Melt curve analysis is a feature of the software and is done after real-time PCR quantification. It gives us the dissociation characteristics of double-stranded DNA in the PCR sample and also the specificity of the PCR reaction. melting temperature or Tm as shown in the graph is a temperature at which 50% of molecules are single stranded. This temperature Tm depends on the nucleotide content, the sequence and the length of the DNA. Let us understand this melt curve analysis with some examples. Here you can see melt curve and melt peak analysis of a number of samples. The different color graphs denote different samples amplified by real-time PCR. The amplification of same DNA template in different samples is done in this PCR. As all the melt curves and peaks are similar, we can be sure that only one product, that is our desired or wanted PCR product, is amplified in this real-time PCR. Now let us see a scenario with non-specific amplification. Here you can see a different pattern of melt curve and melt peak, which suggests non-specific amplification by primer dimers. Now primer dimers have a Tm in the range of 65 to 70 degrees, which is lower than the Tm of the amplicon. Primer dimers form when primers are in excess and there is complementarity between the primers. After studying real-time PCR, let us see what are the advantages of real-time PCR over conventional PCR. The first advantage is that the sensitivity and the specificity of real-time PCR is very high as compared to conventional PCR. The quantitative results are measured in terms of fluorescence value in real-time PCR. Also, the detection method is fluorescence in real-time PCR, whereas in conventional PCR it is agarose gel electrophoresis. The reaction time is also very less in real-time PCR, that is 1 to 2 hours, whereas in conventional PCR it is 3 to 5 hours. Post PCR, we can do a melt curve analysis in real-time PCR. However, we cannot do that in conventional PCR. After studying PCR and real-time PCR, let us see reverse transcription PCR. Reverse transcription is RNA-directed DNA synthesis. When DNA is formed from RNA, it is known as cDNA or complementary DNA. The enzyme used for reverse transcription is known as reverse transcriptase. It is an enzyme found in retroviruses such as HIV. For reverse transcription PCR, enzymes are derived from Meloni strain of murine leukemia virus as well as avian myeloblastosis virus. The reverse transcription takes place at a temperature of 37 to 42 degrees centigrade and DNA primers are used for reverse transcription PCR. In this figure, you can see the procedure of reverse transcription PCR. You can see how the synthesis of first strand of cDNA happens and mRNA is converted into cDNA with the help of reverse transcriptase enzyme. After cDNA is formed, the PCR components form the second strand of cDNA and a double-stranded cDNA is formed. This double-stranded cDNA is amplified in a PCR reaction to get multiple copies of the cDNA. There are three types of priming strategies for reverse transcription PCR. The first one is random primers. Random primers converts all RNA into cDNA. The second strategy is oligo-DT primers. Oligo-DT primers have a tail of T nucleotides which attaches to the poly-A tail of mRNA. So, oligo-DT primers only convert mRNA to cDNA. We can also use a sequence-specific primer and convert specific sequences of RNA to cDNA. Let us see the applications of reverse transcription PCR. Reverse transcription PCR can be used for the quantification of viral antigens such as P24 antigen of HIV, to study the expression levels in cells and tissue and to study the presence or absence of active TB infection. For detection of HIV viral load, RNA is isolated from blood of suspected HIV patients. Reverse transcription PCR is done from RNA of all suspected patients, and PCR for HIV P24 is put using the cDNA formed. You can see in this picture that there are 4 patients, the samples of whom have been run in the 4 lanes of this gel: lane A, lane 1, lane 2, lane 3 and lane 4. You can see a bright band in lane 1 which is suggestive of HIV P24 presence. Now let us understand the applications of PCR as a whole. PCR is a very useful technique in basic biomedical research. It can be used for amplifying specific DNA sequences for cloning and other experiments. It is also very important for diagnosis of genetic diseases. For example, sickle cell anemia can be diagnosed using PCR. We can quantify the normal, carrier or disease state in sickle cell anemia using PCR. PCR is also a very useful technique for infectious biology. Disease diagnosis for infections such as Mycobacterium tuberculosis or HIV infection can be done using PCR. PCR is useful for cancer biology experiments as well, wherein the differential gene expression can be measured using this technique. DNA from fossils can also be analyzed for evolutionary studies using PCR technique. Finally, PCR is very useful for forensic sciences, wherein the techniques like DNA fingerprinting have been developed. Also, PCR and DNA fingerprinting are very useful for addressing paternity disputes. Now let us summarize what we have learned in this module. Polymerase chain reaction is an in vitro enzymatic reaction for DNA amplification. The PCR consists of repetitive cycles of 3 steps: denaturation, annealing and extension. In denaturation, the double-stranded DNA melts into single-stranded DNA. In the second step, that is annealing, primers are used to bind to specific sequences of DNA so that our target DNA is amplified. The third step is extension, in which DNA polymerase, that is Taq polymerase, which is a thermostable DNA polymerase, extends the primers and makes copies of the desired DNA. The primers used in PCR hybridize to the flanking regions of the target DNA. The DNA polymerase is a thermostable DNA polymerase such as Taq polymerase, which has been derived from Thermus aquaticus. Automated temperature cycling can be achieved by using a thermocycler. The thermocycler can increase or decrease temperature in a programmed manner. We have also learned about reverse transcription PCR. This reverse transcription PCR, also known as RT PCR, converts RNA into a complementary DNA strand using reverse transcriptase enzyme. This complementary DNA, also known as cDNA, can be further amplified using the PCR technology. A great advancement over the conventional PCR is the real-time PCR. The real-time PCR can quantify and amplify the DNA fragments simultaneously. The real-time PCR technology uses hydrolysis probes or DNA double-stranded binding dyes such as cyber green dyes to quantitate the DNA formed after each cycle of the PCR reaction. Unlike the conventional PCR, where detection occurs after all the 30 or 35 cycles are complete, in real-time PCR, detection occurs real time, that is after each cycle of amplification. The initial amount of cDNA or mRNA that is used is inversely proportional to the cycle number or CT value in real-time PCR. The real-time PCR is a widely used technology for gene expression profiling, and it has been very useful in molecular diagnosis, forensics, as well as in cancer biology and diagnostics. This brings us to the end of this module. I hope you have understood this module, that is PCR, polymerase chain reaction, and its applications. I hope this module will help you in your studies and clear your concepts about this topic. Thank you.